Very good evening to everyone. It is my pleasure to introduce today's speaker, Dr. Danushka Udayanga, Head, Department of Biosystem Technology, Faculty of Technology, University of Sri Jayawardhanapura. Today he is going to talk about from genes to genome, value advances of using DNA sequence data to study plant pathogenic fungi, which is really valuable topic for day-to-day scientific field. So Dr. Udayanga, first I would like to briefly introduce him. Dr. Udayanga obtained his first degree, BSc Honours degree in Molecular Biology and Biotechnology from Faculty of Science, University of Peradeniya. Right after that, he moved with his higher studies in biotechnology in Lang University in Thailand. During his graduate studies, he traveled to many countries and gained a lot of experience in the molecular phylogenetic studies of pathogenic fungi. With this research finding, he published them in really valuable many prestigious journals. After that, he worked as the postdoctoral research fellow in fungal comparative genomics in United States Department of Agriculture. After serving many years over there, then he moved back to Sri Lanka in 2017 and he joined as the senior lecturer in the Department of Biosystem Technology. So currently he's serving as the head as well as he's serving as the visiting lecturer for many degree programs in different universities as well as in different institutes, as well as he continuing with his excellent research work currently. And with that, he obtained and he identified as the world top 2% scientist in last year. So without further delay, I would like to cordially invite to our distinguished speaker, Dr. Danushka Udayanga, to address this enthusiastic audience. Over to you, Dr. Danushka.
Thank you very much, Dr. Sumudu. I hope you all can hear me, right? Can you hear me?
Yes, Dr. Danushka.
Okay, so can you allow me to share the screen, please? Okay, I hope you all can see my screen. Thanks again, Dr. Sumudu, for a very nice introduction. And also, I would like to thank YSF for inviting me for today's talk. And yeah, although the rain is going on very severely around Colombo, so I think there are possible power failures and all for both of us, for me and you. But still, we will try to continue the talk for today. And for the audience, thanks again for joining today. Today, I'm going to talk quite interesting topic, although there are a large number of people working on plant pathogenic fungi in the world. It's going to be a very hot topic and interesting topic for the people who are actually working on plant pathogenic fungi in different aspects. The topic I have chosen here is from genes to genomes, advances of using DNA sequence data to study plant pathogenic fungi. Basically, my target audience is going to be the YSF members, possibly the young scientists in Sri Lanka, and undergraduates coming in any sort of life science background, biological sciences or agriculture or biosystems technology, as I'm coming from a biosystem technology department. And also, I expect at least a few industry people in Sri Lanka, especially from research institutes, as well as other agriculture related industries. So yeah, without further delay, we will move on with our topic. Right? For those who have less experience in plant pathogenic fungi, I would like to ask, what crosses your mind when we talk about plant pathogenic fungi? For me, there are many things because I'm working on plant pathogenic fungi now for more than 15 years. So I have many things coming into my mind. But there may be a different way that you are feeling when we talk about plant pathogenic fungi. This is something that I feel because a picture coming from my backyard, it's a fruit infection. Right? You all know in Sinhala, we call it as Ugurasa or Flacosia indica plant. So this fruit will get infected with a particular rust fungus. Once the disease is really severe, it will turn into a mummy berry, and it will ultimately fallen down. So we can't eat it basically. So that is something coming to my mind because almost every day I'll see this in my fruit tree in my backyard. Right? There may be many things that you may experience. Right? If you have strawberries purchased recently, and if you have left it in a refrigerator for some time, you may have seen these molds. Those are basically post-harvest problems. Sometimes they can come even from the field, right? And if you are interested in coffee plants get infected with coffee rust. So coffee rust is quite common in plant pathology books, right? If you are a plant pathology student one day, as an undergrad or as a postgrad, you may have heard about the big trouble caused to Sri Lankan coffee cultivation by this coffee rust back in 1800s, actually, 1870 or early 1900s. So it's another very common and destructive plant disease, still kill a lot of coffee plants around the world in coffee growing regions, right? If you are an enthusiast in rice, you will have heard about the diseases in rice, right? This is one of the rice blast infected field, right? So if you are closely looking at your vegetables, right, when they are grown in your home garden or when they are getting old, this kind of anthracnose infections can be commonly seen in chili cultivation. Right? So all of these are correct if you can remind any of those things. Yes, they are plant pathogenic fungi. But normally, do you think the plant pathogenic fungi is a very, very serious problem every day? Actually not. Right? Some plant pathogens come into the plants and they just disappear. Right? So if fungi are infected into a cultivation, you can use fungicides and control it, right? Then it's done. But there are severe situations where the climate is very favorable and the inoculum load is really high. Then these fungi can cause really huge destructions into agricultural crops. Then it will become a really big problem. That's why we should talk about plant pathogenic fungi, right? Although I mentioned it like that, right? There are a few very common examples we can see in plant pathology books, right? So out of many epidemics caused by various plant pathogenic fungi, this is something that I would like to remind you again, right? If you have heard about it. So back in 1940s, there was a big issue in India, in Bengal, right? Possibly due to a fungus infected to all the rice fields. So due to this issue, what we call as Great Bengal famine, which resulted yield losses in rice from 50 to 90%, causing the death of about 2 million people. That's what we can find documented in internet. Right? This is one of such big issue, right? As the people in this era who experience COVID-19, we know that some sort of pandemics are real, right? So humans can be affected, animals can be affected by fungi and viruses, bacteria, or any sort of microorganisms. Similar to that, these agricultural crops also can be severely affected by fungi, causing this kind of disaster. And again, in a publication back in 2012, I found this very interesting information, which I have mentioned to my students many times, right? So it's a hypothetical event. If all these 5 major staple crops like corn, rice, soybean, wheat, and potato were affected simultaneously, approximately 61% of the world population would not have food, because we eat either corn, rice, or in some countries, soybean can be a major source of foods, and wheat, of course, one of the major source of food, and potato is historically really important, and a great famine in Ireland happened due to the fungal-like organisms infected on potato, right? So scientists predict some sort of possible catastrophes in future due to the severe fungal infections. Right? So we should be alert, and we should keep on studying plant pathogenic fungi due to these reasons. Right? Not only because of that, fungi can cause some sort of threats on agricultural trade. The people in agricultural industry, they have experienced this very well, right? There are several high-value crops, some ornamental trees, forest trees in both tropics and temperate regions are currently affected by various fungal infections, right? You will see even in popular news articles, there are certain outbreaks of fungal diseases recorded from many countries. Right? As the people living in tropics, we experience this much more than the other countries because we have a very favorable environment for the growth of fungi. Right? So fungal diseases with quarantine concern could cause various issues in the exchange of plant-based products and materials across the borders. Right? When we try to export some plant products, there could be issues due to the contamination of various fungi on them, because many of the countries, they are scared of coming invasive fungi into their land. Right? So as an island, we also very cautious about our biodiversity. So our quarantine officials usually working tirelessly to stop that kind of infection to our native plants and crops. Right? So these are a few examples coming into my mind. The grains of wheat infected by kernel bun. So wheat is a big player in global agricultural trade. So if there is a fungus like this, nobody will accept that kind of shipment into their country. Right? So blister blight in tea is very common even in Sri Lanka and India, especially in these two countries. So that is one of the major threats in our tea cultivation, right? Although it's not going with the tea production, it can eventually kill the tea plants when it's severely infected, right? And in our day-to-day life, if you are wandering around markets or if you are buying fruits, you may have seen anthracnose diseases of various fruits, not only in mango, right? So nobody would like to buy a fruit with black color patches. We don't like it, right? So it's obviously a problem for the agricultural trade. And there is a hot topic going on in Sri Lanka these days. If you closely look at this news segment that I have extracted from Economy Next web journal, right? So they report that Sri Lankan rubber output to drop 15% in 2023 in deepening the crisis. This crisis is all about a fungal infection. Right? The circular spot of rubber leaves have caused much damage to the Sri Lankan rubber cultivation nowadays. So this first epidemic reported in Malaysia back in 2017 and 18, and the disease then spread to Indonesia, devastating many rubber plantations, and there's an ongoing outbreak in Sri Lanka. So this news article clearly describes the danger of this particular outbreak, right? The fungus might be Pestalozziopsis or Neopestalozziopsis like fungus. So there are a lot of work going on in the country regarding possible control measures to this circular leaf spot of rubber. Right? So that is one of the very interesting example for us to talk about this topic today more than any other day. That's why I particularly selected this one, right? So we can give like hundreds of examples for various fungal infections, but they will be highlighted when something happened like that, especially a threat to our economy. Usually, studying plant pathogenic fungi needs a quite combined, consolidated approach. Right? We cannot study plant pathogenic fungi before you are getting a good background knowledge and also good guidance to study them. There are a few reasons, right? So as a teacher for like 6-7 years now, right? So I have seen these three examples or three points to show that why plant pathogenic fungi should be studied and why it should be very carefully done, right? So plant pathogenic fungi are very much taxonomically diverse, right? Although we can show a few examples based on our knowledge, we can't tell the number of different fungi infect to even one crop plant, right? That can be an enormous number sometimes. They are taxonomically very diverse. They belong in different classes, families, and phyla, right? And also, these fungi are morphologically very plastic, right? They can be looks different sometimes, even on one host. And also, the same fungus can infect many different plants, right? That can mislead the people who study plant pathogenic fungi. And also, it's really difficult to define species. Just like you are identifying a plant or an animal or any other very larger organism, it is really difficult to identify a microorganism. Among them, fungi are again difficult more and more, right? There are more than one fungus involved in one disease, right? In that case, we will find many species complexes associated with one disease. That is again a complex situation when it comes to studying plant pathogenic fungi, right? This is why we should be very thorough when we learn and when we do research on plant pathogenic fungi, and also we should be able to use more technologies to solve these problems and study them very carefully. So in my research career, always I would like to talk about the molecular identification of plant pathogenic fungi or DNA sequence dependent identification of fungi. Yeah, okay, power drop, but I think still you all can hear me. That's so. Glad that you all can hear me at least. You continue like this, right? If you all can hear me. Is that okay to
Yes.
continue?
Yes. From this
Okay.
slide, please.
Yeah. So this is one of the favorite thing that I would like to talk. That is the DNA sequence dependent identification of fungi. So usually in past, we identify all these fungi based on morphology, or sometimes maybe based on host association. Right? If we isolate the fungus from a particular host, scientists tend to give a name based on that particular host. That is one possibility. Right? Otherwise, they study the morphology really carefully by looking at their spores and their reproductive structures, then they name the fungus. But with the advancement of molecular technologies, mycologists basically, and also plant pathologists, they started using DNA sequence data to identify plant pathogenic fungi. They selected a suitable DNA sequence which can act as a molecular marker. Right? So there are so many different kinds of primary and secondary markers we can use based on literature. Right? Among them, yeah, so those who are not very familiar with this diagram, the first diagram I have shown is a sample of a DNA sequence we could get from a sequencing company if you ask for DNA sequencing, right? If you have a DNA sequencer in your laboratory, this is the output you will get once you have sequenced a segment of genes in a fungus or any other living organisms, right? Below, I have shown two approaches where we can use this kind of DNA sequence. One thing, you can use it as a DNA barcode, right? Right after the DNA sequence is generated, you can make a quality control, and then you can use that as a barcode to directly identify the fungus using the public data. Otherwise, you can use these DNA sequences to construct phylogenetic trees. Right? So basically, we construct phylogenetic trees to identify the evolution very clearly, which give much more information compared to the straightforward barcode. Those who have learned about fungi and fungal systematics, right, you may have heard about the ITS region, that is the standard fungal barcode. I just want to show you, right, this is a kind of a gene region which has mostly sequenced and available in public databases for comparison. For today's research, we mostly recognize ITS as a quite good barcode region to identify fungi. So once we get an unknown fungus, if it is impossible to identify morphologically, then we have the option to do a DNA sequence, particularly for ITS region, and try to identify it by comparing the public databases. This is one of the diagrams I have generated for a research paper some time ago, which basically outline how a DNA sequence can be used in fungal identification, right? Say, for example, if you start with the fungus on a mango fruit, anthracnose-causing fungi, right? You can isolate them, you can observe them under microscope, you can culture them, but still we are unable to name them accurately, right? If you are a really experienced mycologist or plant pathologist, you should be able to name it, right? But still that name can go wrong. Then there's a role for a molecular mycologist to use DNA sequences and identify them. So you can sequence a region like ITS, translation elongation factor 1 alpha or beta tubulin kind of region, and you can compare them with the public databases like GenBank, or you can construct a phylogenetic tree with the guidance of an experienced person if you are not experienced with phylogenetic work. What I need to highlight here, why do we need to make phylogenetic tree actually when you have a sequence analytics by BLAST? This is a very common question I'm getting from many of my past students, right? So you can use a DNA sequence to identify a fungus, but still we can make a phylogenetic tree to make it much more convincing to the audience, to the people who read the research papers and use this research, because phylogenetic trees shows the evolution very accurately, right? That is hypothesis, but still it will give much more information about that particular fungus. Right? So why it is recommended to use several genes rather than using one gene? That is again a very, very common question we asked as students when we were doing our postgrads, right? So several unlinked genes can combine and make phylogenetic trees to make that phylogenetic hypothesis much more accurate. That's why we tend to use multi-gene phylogenies or many genes in the phylogenetic work. So if you are into using genes or DNA sequencing to identify fungi, there are a few things when you need to select the gene to sequence, right? That can be the level of taxonomic hierarchy you are working on. If you are looking at species, then you have to be very careful and choose the right gene that can give a separation at the species level. If you are working at the genus level or maybe phylum level or kingdom level, then you have to be very cautious and select the right gene region to sequence. Then we have to look at your purpose, whether it's just barcoding or the phylogenetic reconstruction. And sometimes you have to look at the complexity of that particular fungus group. Some fungi are easily identifiable, but some are very difficult. Right? And also you have to look at the current status of that particular genus and also the speciation pattern. And the knowledge of current classification is really important. Right? If you are not a very experienced person in plant pathology or mycology, the best thing to ask the expert mycologist before you move on to a project based on using DNA sequences to identify fungi. Right? This is few examples that I can show you, which is based on my group over past 2 to 3 years. Right? We tried to identify the fungus which caused this anthracnose twisted disease of onion, right, which was an outbreak happened back in 2020 around the onion growing regions in Sri Lanka, around Matale, Tambulla and the vicinity. Right? So we were able to identify this fungus as Corydotrichum siamens, and this research was published back in 2021 in Australasian Plant Disease Notes as a small disease note. But you can see this particular simple experiment or simple research work give very important information for our agriculture. And this is one of the work that I have done when I was doing my postdocs in USA. Right? So this strawberry leaf blight fungus was a very common fungus infecting strawberries, but unfortunately the name was quite uncertain, although people called it as Phomopsis obscurans earlier. Right? So our study revealed that actually this is not a Phomopsis, and it belongs in another new genus. So we described the new genus as Paraphomopsis, and the species was named as Paraphomopsis obscurans based on molecular data. Right? This is one of the two of the examples that I can quickly give from our recent work. Right, so you can see how these information are going to be impacting to the agricultural and industrial community when they are nicely done. But although we can use genes to characterize fungi, there are certain limitations. Right? These insufficient phylogenetic information or sometimes gene-specific noise or problems of using single or a few loci or few DNA sequences often generate disagreements or incongruent phylogenies. And also, there's no information about biology or potential virulence when we just identify them using single genes. Right? So obviously, we need more data through genomes, that is more and more genes, possibly the whole genomes present in the plant pathogenic fungi as a solution to give more data in research, right? That's why we tend to work on genomic stuff. And most of the time when there are students who want to move for higher studies after their undergrads, right? They would like to move for genomic studies, maybe in the human disease side or human biology, or sometimes zoological studies or botanical studies. But there are people who really interested in the genomics of microorganisms, including fungi. But unfortunately, they have very less exposure in our country. But I would like to remind all the students who have joined here today, there are plenty of opportunities for you to learn. Right? So in case of plant pathogenic fungi, in this era of genomics, there are endless opportunities in research. Right? We can sequence fungal genomes from various fungal groups, but various scientists use the fungal genomic data for different purposes. Right? If there are scientists who are working on the metabolites of fungi, they do the genomics to study their metabolites much more clearly. But plant pathologists and mycologists, they do genomes to identify the biology of infection and the severity of the infection of pathogen, and also to predict possible outbreaks, possible epidemics, which can cause by that particular plant pathogenic species in future. So the DNA sequencing technologies have rapidly evolved. Right? So if you have heard about the DNA sequencing technologies, in past, we used mostly this first generation DNA sequencing, what we call as Sanger sequencing or Maxwell and Gilbert, or the chain termination methods, which generated 500 to 1,000 base pair long fragments. But when it comes to the next generation or the second generation sequencing, we have many platforms provided by biotech companies. Right? Few examples I have given in here: 454 pyro sequencing, Solexa, Iron Torrent, and more popular Illumina sequencing. Those platforms are second generation sequencing. But nowadays, even the undergraduate students, they talk about the third generation sequencing, including PacBio and the Oxford Nanopore technologies, where we can generate very long fragments of DNA of any living organisms that you would like to work on. So this evolution of sequencing technologies has a direct impact on plant pathological studies, especially to learn about fungi more and more. Since the next generation sequencing is a highlight, right? There are a variety of methods which can generate thousands or millions of sequence in parallel in a single experiments in next generation sequencing platforms. Like 5 to 10 years ago, maybe in Sri Lanka, we didn't see any NGS platforms. But nowadays, I think several institutions have this kind of next generation sequencing machines, and they are using them for various purposes. So in case of NGS, preparation of a sequencing library is a distinctive feature. And also this library preparation method is going to be unique from one platform to the other, right? If you are using Illumina platform, there are several of Illumina sectors in that company, which they offer for genome sequencing. So when it comes to sequencing of plant pathogenic fungi, what do you think? Right. So this paper I have extracted the diagram is coming from 2017. It was published in IMA Fungus. So they have studies like 1090 fungal species, and they have classified the nature of those fungal genomes. And so these scientists indicate, so they show that pathogens comprise the largest category with available genome sequences are pathogens. Among them, there are many plant pathogens sequenced and available in public databases. Right? So this is in 2019. So now it is 2023. Of course, the statistics have improved. There are many plant pathogen genomes available in public databases. Right? So it is possible to use this genomic data in your analysis, in your research work, and also if you are in industry or research institutes working on various pathogenic fungi, there are plenty of data available in the databases, right? Only thing, you have to learn the techniques to use them, and you have to be aware of them and make use of them for your purpose, right? This is one of the popular website for the people who sequence plant pathogenic fungal genomes or all the fungal genomes. So Joint Genome Institution offer this particular website known as MycoPerson. There are a lot of fungal genomes available publicly, right? If you need to study fungal genomes, you can go ahead and download them and use in your research, right? That is for your information, those who are not very familiar with fungal genomic resources available in internet, right? There was a project initiated back in 2010 or 2012, that is 1000 fungal genomes project. So I'm pretty sure now there are more than 1000 fungal genomes are sequenced and available in public databases. So what actually fungal genomes reveal compared to the genes that we talked earlier? If we have the genome data available, it gives better understanding of pathogenicity, lifestyle, and the genome evolution of that particular pathogen through the downstream analysis of structural, functional, and comparative genomic analysis, right? So if we are working on a particular emerging pathogen or a disease, right, we can obtain the genome sequencing of that particular pathogen. So nowadays, plenty of sources are available, plenty of platforms are available to generate genome sequences, right? Then we can study the evolution, infection, biology, virulence, and also effectors and toxins based on the mining of that genome. Those data can be effectively used in disease management practices. Although it's a long pathway, the most interesting thing about genome-driven data is they are very accurate and reliable, because that is coming from the DNA of that particular living organism. Again, if you are a fungal enthusiast or genomic enthusiast who would like to move to this direction in future, as young scientists, as young undergraduates, right, you should know at least the rough background or the technical knowledge for this particular genome project, right? So if you are looking into a fungus which may be very interesting or which can cause a severe disease, right? Isolation of that fungus is really important, right? If it is a culturable fungus, then you can verify the identity using simple fungal barcoding or multi-gene phylogenetic analysis, right? Then you can select an accurate sequencing strategy or next generation sequencing platform to sequence the genome. So if you want to obtain the genome sequence data, the first thing to do is to obtain high quality DNA. This is a must. So nowadays, there are genome sequencing platforms, even from a little amount of DNA, you can get the genome, but always it is recommended to use high quality DNA in genome project. Right? So in parallel to the genome sequencing, we have to do transcriptomes because that is important for the analysis or assembly of the genome. Then you need high quality RNA. Then you have to prepare the libraries if you have access to the genome sequencing facility in your institutes. Otherwise, you have to send these DNA and RNA to the biotech companies for sequencing. Right? Then the equipments and the logistics are really important because we need money for DNA sequencing or genome sequencing. Then the quality control of genome data and genome assembly will generate a draft genome that you need for the downstream analysis. But having a genome will not solve any problem. Then the game starts from that point. You have to learn downstream analysis of genome data. Right? We take in Sri Lanka, we have very few people who have experience with analysis of genome sequences, right? So that's why I again and again stress all these young people who are interested in when you go to foreign countries, learn the new things and bring them back to the country. That is something that I have told many of my students, right? So in case of downstream analysis, we can do many things using an available genome sequence of a fungus. You can do the assembly, then you can annotate the genome, then you can obtain the selection analysis. So species specific genes, if you need molecular markers, you can do genome-wide phylogenomic reconstructions. Right? You can study the evolution and you can study the gene flow and the reproductory mood and demography, right? All those things can be studied. Right? There are few important points mentioned in this particular diagram that I extracted from 2017 publication in Frontiers in Plant Science. Right? You can see the questions below. Right? Those questions highlight how much the fungal plant pathogen genomes can reveal in terms of basic science and applied sciences, right? So what is the genetic basis of specialization? And if there's a hybridization going on, we can reveal that using genome analysis. Then we can determine the horizontal gene transfer if there are such evidences and so on, right? So if you are interested in, I highly recommend you to go to this particular publication and read it. That particular review paper highlight many of the things that you should know when we are into studying plant pathogenic fungi in the genomic era. So these questions are really important. That's why I have highlighted for you, right? So with the time, I'm not going to go through all diagram. Keep on moving. In the same publication, there's a very nice diagram which highlight the genomic basis of pathogenicity, lifestyle, and host adaptation of various plant pathogens, right? So if I show you one, you can see in case of pathogenic and non-pathogenic comparison in terms of the genome size, right, that is obviously different due to the presence of various transposable elements more in the pathogenic strains or pathogenic species. Right? When you look at the nature or the infection nature and the biology of pathogens, there can be biotrophic, hemibiotrophic, and necrotrophic ones. So the higher amount of cell wall degrading enzymes are usually present in necrotrophic plant pathogens compared to the biotrophic ones which infect the living tissues. And also in case of host specialization, when the fungal plant pathogens become specialized into one host, right, there are indications in the genomes like signal peptides may present compared to the non-specialized one. And when the fungal pathogens jump from one host to the other, right, they may carry the genes like tox A in this particular example, right? So these footprints of genomes can be present in both genomes due to the host jump. So there are many information we can reveal the structural, functional, and comparative analysis of fungal genomes. Right, the amount of data that you can obtain, the amount of information that you can obtain by doing a genome work is enormous. Right, so in recent past, as a postdoc, I have done some of the genome sequences when I was in USA, and the person, as a person who have done a lot of phylogenetic studies, when you become into the genomic world, that is going to be really exciting for the person who is enthusiastic in that field. That's why I always encouraged our students to go ahead and learn these cutting edge technologies and bring them back to apply in the country in our research too. Finally, I would like to tell something interesting again. We can use these metagenomic tools or the environmental sequencing tools also in studying plant diseases. In case of environmental sequences, unlike a single genome sequence, here we obtain the environmental samples, which can be soil, which can be a tissue from the environment, which can be a water sample, and we extract the all DNA present in that particular environmental sample, and we can do either diversity analysis or we can do a functional analysis for the genes or the DNA present in that particular environmental samples. Right? So that again changed the statistics of what we know about plant diseases in future. Right? For example, nowadays, if we observe a plant disease in a leaf or fruit, right, we are going to isolate it very carefully. Most of the time, we will end up with 1 or 2 fungi infecting that particular specimen. But if you do environmental sequencing, there may be amazing things, there may be groundbreaking findings in the environmental sequencing data, because a lot more unknown fungi can be associated with the same specimen, right, due to various reasons like their unculturable nature or the amount of the fungi present in the tissue, right? We may not detect them by simple methods, right? So this particular nice diagram I obtained from a publication in Scientific Reports back in 2022 highlight how metagenomic detection of a leaf blight pathogen, Calonectria pseudonaviculata, can be done. Right? So if you go through the diagram carefully, you can see there's a leaf. The common methods, you can just take the infected leaf to the laboratory, then you can look at it very carefully and you can isolate them, culture them, and do the DNA sequencing and identify. That is one conventional method we discussed, right? But you can go extra level and you can use this specimen itself and crush it and take all the eDNA or environmental DNA to your extract, and you can make a environmental sequencing library or metagenomic library and use that library in a next generation sequencing platform, either nanopore sequencing or Illumina sequencing platform. Right? Then you will end up with a lot of DNA sequences, right? Among them, you can of course omit the plant's genomic sequences from that, and then characterize the other fungi or maybe bacteria present in the specimen, right? Again, this will change the way we look at plant pathogenic fungi and their diversity and the way we study in future, right? So of course, genomics and metagenomic tools are going to be the cutting edge tools that you may use if you go into these fields, especially in other countries. And also, even in our countries, I hope our country, I hope that the way that we look at research will be will be different in recent years with the development of technology. But still, there are few bottlenecks of this new approach of plant disease projects, right? For us, like in Sri Lanka, it can be the lack of funding, sometimes the lack of sequencing technologies, and also lack of expertise in analysis, right? So if you need to work on genome sequences, it is highly recommended to get a very good training with an experienced person. And that can be done with the collaboration. And also, due to the lack of general awareness and sense, people are still not using these technologies. So that's why I would like to educate all this young community with the basics of using genes and genomes to study plant pathogenic fungi more and more. That will come to end of my talk today. Right before that, I have this take-home message to all of you for these young minds who can be the fungal enthusiast or life science enthusiast who would like to come to the field in future. So in this era of genomics, we have significant tools and technologies to deal with the plant disease causing fungi, and we must apply them more actively and aggressively for the betterment of mankind, ensuring global food security and sustainable agricultural trade. This is what I want to highlight from today's talk. So I'm pretty sure you will get some sort of idea, although the person who may not have a big exposure to genes and genomes, this is not going to be best as their first talk, but I think that this will make your idea to work on fungi in future, and you can make a start from this point if you are interested in. And I wish you good luck for those who are looking forward to do the higher studies in near future. Yeah, from that, I would like to say thank you again to YSF and Dr. Sumudu for inviting me and the audience who came here today.